Thank you. Thanks so much for the invitation. All right, I'm going to try and share my screen here. Okay, wonderful. Thank you very much to the organizers. I really love that this is a student and trainee organized conference, and I'm quite honored to be here virtually. I'm in New York. I've been up since about 3 o'clock in the morning my time, so bear with me if I'm a little woozy during the talk here. So I'm going to tell you, I'm an associate professor at Cold Spring Harbor Lab in the quantitative biology department. I'm going to tell you about what my lab does to do with transposon genomics and how these things are regulated and how they might contribute to neurodegenerative diseases. I'm also going to put a timer on for myself so I keep on time here. All right, so just a disclosure first, I'm on a scientific advisory board of a company called Transposon Therapeutics, which I have to say before every talk. So just to remind you, I'm sure everybody here knows what transposable elements are, but just as a little reminder, these are mobile genetic elements, and the retrotransposon subset of it, which is most in most animal genomes, these can make a copy of themselves. That RNA copy is converted back into DNA, and that copy is inserted somewhere else in the genome, right? So these things have been very active, very proliferative. They've made many, many copies of themselves, and there's many, many copies of these transposable elements in our genome, the human genome. But in addition, most plant and animal genomes also contain transposable elements as well, which is why these were originally discovered in maize, the corn plant maize, by Barbara McClintock in the early 1940s, right here at Cold Spring Harbor. So, okay. There we go. Transposition, mobility, the ability to move throughout the genome. This is what transposons are famous for, but this is not the only thing they can do. So they can alter the function of genes that they hop into, either by breaking that gene or by giving it a new function by inserting protein coding domains within that gene, for example. In addition, they don't have to be inserted in a gene. They can be inserted adjacent to a gene and regulate the expression of that gene, either by the host trying to silence the transposon and having that spread to nearby regions, or having the promoter element of that transposon turn on adjacent gene regions. That's another thing that's known to occur downstream of transposon activity. And the spread of silencing on the bottom is the one that I showed you there as well. So most of these have been detrimental things that are due to the presence of transposons. There's also another thing that we don't talk about often in terms of what transposable elements do, and that's to create potentially immunogenic byproducts, right? So I told you that these transposons make RNA copies of themselves, and then they convert that RNA to cDNA that happens out in the cytoplasm. So those double-stranded RNAs or double-stranded RNA regions and the cDNAs that are produced from that, those can often alert innate immune sensors that are looking for viruses and other pathogens. And these things can look viral-like, enough so that they alert these innate immune sensors, either the RNA sensors or the DNA sensors, and promote pro-inflammatory pathways downstream of that. That's been shown, I think, most in Aicardi-Gutierrez syndrome, certain kinds of cancer, and other systems as well. But this is another thing that we have to keep in mind in terms of what can go wrong when transposons are aberrantly active. So, okay, and in one of my favorite demonstrations of this recently is from a colleague of mine at Lund University named Johan Jacobsen, and in his lab, they've shown that just upregulating transposons themselves through knocking out one of the regulators, TRIM28, was enough to induce an inflammatory response, meaning activated microglia in the brains of those mice that had desilenced retrotransposons. And so this is in the absence of any other disease pathology. And I think that's a really nice demonstration that transposons actually are sufficient to induce these innate immune pathways and these pro-inflammatory responses in the CNS. Just to show a teeny tiny bit of the data, this is Johan's data. It was predominantly transposons and very few other genes that were dysregulated downstream of TRIM28. That's shown here. And then you can see that in the adults where these transposons were highly expressed, it was mostly LTR retrotransposons, that induced pro-inflammatory microglia that are hypertrophic and that are expressing high levels of IBO1, this pro-inflammatory marker on activated microglia. And I think that was a really nice paper that came out last year from Johan's lab. We're continuing work on this. So I've told you all of these bad things about what transposons can do. They're in every plant and animal genome for the most part. In some species like maize, they outnumber the amount of the genome covered by host genes. So in maize, it's 80% transposons and only 20% other stuff in the maize genome. For humans, that's about 50%. And they hop around making copies themselves and otherwise causing havoc when they're active. And so you might wonder, if these things are so bad, why don't we hear about these all the time? And why are we mostly okay? So I'm going to take a little bit to give us some of the answers that we know about those two questions. So one reason that we don't hear about transposons as often as we might is that they're often discarded from most genomic sequencing studies because they are a pain to work with. And what I mean by that more specifically in terms of them being difficult to work with is that, for example, the L1 retrotransposon subfamily covers 17% of the entire genome in the human genome. That's made up of about almost 1 million copies of this 6 kb line 1 element, some of which are a little bit smaller than that. But out of those 1 million copies of L1, only about maybe 100 to 200 of them are fully intact functional versions of the L1HS retrotransposon that can make new copies of itself. So trying to tell the difference in a short read sequencing assay or even a longer read sequencing assay between one of these 100 full-length competent transposons and the 1 million other copies of these in the genome is computationally a very difficult problem. And so for the most part, it's been ignored. So my lab and many others have been working on tools to handle this problem so that we can really look at transposons properly and ask what exactly they can and do in relation to human disease. So many excellent labs have been working on these for years, but only really recently have we had better tools to handle these at a molecular and a genomic level, right? So better technologies, meaning longer reads with fewer errors in them, better software that includes statistical read assignments rather than just discarding any ambiguously mapped reads, as well as more data about transposons that are present, maybe not in the reference genome, but that differ in different species and different individuals of that species. So in other words, more TE-omes, right? So what my lab has been doing has been working on something called the TE Toolkit. This is a suite of genomic software tools that are for standard genomics assays like RNA-seq, ChIP-seq, things like that. So I'm going to go through a cartoon example of how these work, because I think this is the best way to describe it. So I've told you before that the biggest problem with transposons is that the reads might map to one transposon copy. Let's say that's copy A over here. But there are similar regions within A that could map to many, many, many other places, right? So that's symbolized by this little red line that could be in any one of these 4 other copies. Let's pretend there's only 4 copies of this particular transposon in the genome, right? And if you were just going by, should this read map here, 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 or here, you might not be able to tell the difference between these 3, right? So what my lab has realized is that there's more information here than just how well each individual read maps. Because for an actual expressed retrotransposon, reads will pile up along the full length of this thing and not just along regions of sequence similarity to other copies, right? So we can use that to try and figure out which one of these reads are coming from real expressed loci and which ones are just mismapping to other regions of sequence similarity. So what we use for that is called an expectation maximization loop. It's just a little maximum likelihood framework that runs back and forth, redistributing the ambiguously mapped reads and then recalculating the abundances until those abundances don't change when we keep moving reads. And we go around these loops typically about 10 times before the reads settle down. And you might, for this particular one, decide that these 2 loci here are both expressed, but these other 2 loci, B and C here, are not. Right? And that's how this works. And we can do this for every transposon at every locus in the genome. And we can actually get pretty good estimates of which loci are actually active in any given cell type and context. And so when I say good estimates, we're going to measure this in a couple ways. So one way is we're going to take simulated reads. So we just simulate expression from a whole bunch of different loci in the genome, chop those up into little pieces that are about the length of a sequencing read, add some sequencing errors and other things to make it look like a real RNA-seq assay, and then try to map them back to the original locus they came from and ask, did we get it right? Right? And it turns out that for the youngest transposable elements that are most similar to each other. This is a hard problem. And if you take just unique reads only, that's what these ones down here are doing, you're going to underestimate how much expression is actually really coming from these transposable elements. But if you actually take into account those ambiguous reads and statistically redistribute them to the most likely locus of origin, that's this red dot here, that's the software my group built to e-transcripts, you can actually get pretty close to the ground truth, which is the black dots here, right? And we've been able to get accuracy rates that are above 90% for most of these, and even for the hardest transposable elements that are young and still close to their consensus. We can go a little bit further and we can measure this in real data. So we can take digital droplet PCR data that's specific to different loci, and we can take RNA-seq data from exactly the same samples, exactly the same RNA prep, and then we can ask, do these two things match up? Did we correctly analyze that RNA sequencing data such that it matches what the locus-specific digital droplet PCR will match? Right? And if we're right, then we should lie right along this line here, the x equals y line. And what you can see is that once again, TE transcripts, even though it's getting a few loci long, these are really young ones here, we're actually very close to the x equals y line and probably about as close as you can ever get qPCR and RNA-seq data to match up, right? And generally other algorithms will under or overestimate these things by a little bit. Okay, so now that that software is in place and working, we've been using that in my lab quite a bit for a lot of the studies that we look at. And in addition, we've taken that same basic idea of using these EM loops to statistically redistribute reads, and we've been applying them to basically every other genomics assay that my lab is interested in, right? So if my lab is interested in how transposons are controlled transcriptionally by transcriptional repressor networks, we might be interested in using TE peaks to use those EM reads to look in the heterochromatic regions of the genome for ChIP-seq reads. And we've done the same thing for small RNAs, for locus-specific analysis, and for single-cell data. And I'm going to tell you a bit more about these as we go along, because each one of these assays has its own quirks, and you have to get specific to the quirks of that assay in order to do this well. Okay, so now I'm going to go back to that question I asked you before, which is why don't we hear more about transposons? And so the first answer that I told you is that they're hard to look at. It's hard to include these in genomics assays. The second answer to this question, why are we mostly okay? In other words, why aren't our genomes shredded into bits all the time by active transposons mutating things? Now there's a different answer that's more biologically driven, and that's what I'm going to tell you about next. Right? So it turns out that the vast majority of transposable elements present in most genomes are non-functional copies. They're old, they've accumulated mutations, they're no longer full length, they can no longer actively transpose and make new copies of themselves. They're not quite harmful enough to be fully driven out yet, but they're also maybe not quite recycled into something useful either. Because one of the things I won't be talking about during this talk is how transposons have been extracted and recycled by the genome to create beneficial functions for its host. Right. So I'm going to just tell you that that is definitely a thing that happens, and then leave it there, and then talk for the rest of this talk about what happens when these things are causing trouble. Okay. But one of the reasons they don't cause as much trouble as they might is that many of them are essentially fossilized remains of once active transposons. Okay, so then the second half of the answer to this question is that your cells devote enormous resources to preventing transposable element activity, right? So there are core factors at each stage of the transposon life cycle that are blocking activity from these loci. So there are things that keep the genome closed, keep heterochromatin intact, things that block transcription, transcriptional repressors here that block transcription of these elements. Once RNAs are generated from a given loci, if they escape this first bit, there are specific factors that look and surveil for transposon RNA products, for example, and degrade that transposon RNA before it can be converted to cDNA and inserted somewhere else. And again, there's core factors that block that insertion process, right? And so this is another reason, given how many resources are here to devote it, why these transposons aren't active all the time in all contexts. And while we know a lot about these core factors and how they function, one of the things that we've realized is that there are many more factors that are involved in this process. And the reason that we don't know about, and the reason that we know that there's more factors we don't know about, is we keep finding new ones. And that's one of the things that my lab has been doing as well. right? And so in particular, I'm going to tell you about a couple factors. There's 3 genes here in particular that I'm talking about: tau, C9orf72, and TDP43, all of which have been recently described as playing a role in transposable element silencing. And the reason I'm highlighting these particular 3 genes and not others is these all also play roles in neurodegenerative diseases. TDP43 in the case of ALS, tau in the case of FTD, and C9orf72 in the case of FTD and ALS and Alzheimer's as well. All of these have been shown to be present as aggregates in Alzheimer's as well. Okay, so I'm going to focus now on TDP43. That's what my lab works on. And I'm going to show you a little bit of the data that we use to figure out that TDP43 is playing a role. So let me show you that. So what is TDP43? This is a protein that's normally present in the nucleus of most cells, but especially in motor neurons. It plays roles in gene regulation there, alternative splicing, and trafficking out of the nucleus and into the cytoplasm. In TDP43 proteinopathy diseases, so diseases associated with TDP43 dysfunction, that includes ALS, Lou Gehrig's disease, and motor neuron diseases, as well as frontotemporal dementia and Alzheimer's. TDP43 in its pre-pathological state gets stuck out in the cytoplasm. So it scatters out here to the cytoplasm and is no longer performing its normal roles in the nucleus and gene regulation. And eventually, at the end stage of the disease, you'll get very, very large accumulations of TDP43 protein that might exist in these fibrils or in these very large aggregates. And 95% of ALS patients show this TDP43 pathology. This is a hallmark of the disease. This is how they determined that you definitely had ALS. Okay, so why does my lab think that TDP43 interacts with transposons? Well, one of the other things that we know about TDP43 is that it's an RNA binding protein. And we had some genetic evidence that it was interacting with transposons, but we didn't know how. And so we thought that the most likely scenario was that maybe it was binding their RNA. That was one of the processes I told you about by which factors might silence transposons. So we use ClipSeq assays to pull TDP43 out of cells, this is in healthy functioning normal cells, and ask whether transposons were a part of its targets. Okay, so we did that. We used those ClipSeq assays. This is just to show you that the ClipSeq assays worked and that they're fairly clean. So these are those ClipSeq peaks piling up on one of TDP43's known targets. This is the gene FUS, a gene for which TDP43 regulates its alternative splicing and mRNA stability, right? And you can see that the peaks pile up really just at that ClipSeq site. And there's a little TG motif that TDP43 recognizes and binds right under that ClipSeq site. And it's pretty clean. It's not piling up elsewhere along the genome, right? But in addition to that, we started looking at transposon loci to see if the ClipSeq peaks would pile up there as well. And that again turned out to be true. So each one of these little blocks here is a line 1 element present in the genome. You can see in the RNA-seq data that these line 1 elements are all expressed in this particular cell type. And you can see there's a little TDP43 ClipSeq peak here, 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 and here in each of these line elements. And underneath each one of those little peaks is one of those TG binding motifs. So it really does seem to be recognizing its targets, whether those targets be transposons or genes. And it turns out that transposons are a significant fraction of all of the targets of TDP43 proteins. So here, they're about 20% of all targets are in these transposable element sites. So, okay, so now we know TDP43 definitely binds the RNA of transposons. The next thing we did is to knock TDP43 down, right? To ask whether or not that matters, whether or not that does anything to the cell. And what you can see is that when you knock TDP43 down and look at its gene targets, some go up, some go down. That's typically what happens whenever you knock out your favorite gene. But for the transposons, they all went up. Pretty much every transposon that was expressed in the cell, except a few here, went up in the absence of functional TDP43 protein, suggesting this is a general silencing factor for retrotransposons. And almost all of these were also found in that ClipSeq data, suggesting these are direct targets, right? So that's what let us put TDP43 here. There's now several other labs that have also demonstrated that TDP43 has a role in transposon regulation. And what we're now trying to figure out is exactly what it's doing, and also whether or not that's clinically relevant for patients with TDP43 proteinopathies. So at this point, in order to ask that second question about clinical relevance, I collaborated with a wonderful professor at Columbia in the New York Genome Center named Hammell who was running a very big, large-scale patient profiling study for ALS and FTD patients with TDP43 pathology. So we had about 178 samples for which we had both whole genome sequencing and RNA sequencing data from the affected tissue and motor cortex, where we could ask, do they have transposon desilencing in those tissues? And this was the largest study for ALS at the time, which was a couple of years ago now. So, okay, I expected that what we would find would be that all ALS patients would have TDP43 pathology and the controls wouldn't, and would be simple like that. It turns out that's not quite what we saw. Instead, it turned out that the ALS patients segregated into 3 separate groups, right? And all of these groups were different from each other and also different from the controls. And you can see these 3 separate groups based on a heat map of the genes that mark these clusters, as well as here on a PCA plot where we're plotting how these things separate out. And my group used non-negative matrix factorization as the de novo clustering method for this, but I'd be happy to talk about that later if you want to. So, okay, so what about transposons though? Are they relevant? Well, it turns out that transposable elements, the elevated expression from them is the defining marker of the first group, right? So 20% of the samples fall in this ALS-TE group, where TEs, like these young line 1 elements and SV elements, were strong markers straight out of the NMF algorithms with no seeding or supervision. And in addition, they also had dysregulation of other TDP43 gene targets. And when we went back and scanned those slides to look for TDP43 pathology, these were the patients that had the densest amount of phospho-TDP43 aggregate pathology in cortex in adjacent slices of tissue to the ones we'd used for profiling by RNA-seq, suggesting that transposable element expression and TDP43 dysfunction are really tightly linked and are what's driving the transcriptome profiles of this group. And in addition to the motor cortex samples, which is the infected tissue for ALS patients, we also had frontal cortex samples for some of them, and we could go back in and again ask that same question: does expression of transposable elements here correlate with pathology of the tissue? And that was again true, even if they hadn't been cross-diagnosed with frontotemporal dementia, which is typically what you see when you have strong aggregates in frontal cortex. Okay, so I'm going to talk to you very, very briefly about the other 2 subtypes, because these turned out to be very interesting as well. So there was a second subtype of ALS patients for whom the predominant markers were things related to oxidative stress, real oxidative stress markers like OXR1 and thyroidoxin, in addition to the sort of classical gene SOD1. ER stress and autophagy markers, and general mitochondrial dysfunction. That was what was going on in this group here that did not show transposable element elevation, but did show other markers from pathways known to be affected in ALS disease. And finally, there's this last group, the ALS glia group, where we were seeing genes of pro-inflammatory pathways, activated glial markers from both microglia and astrocytes. And again, we played that trick where we went back in and stained adjacent slices of tissue from adjacent pieces to what we profiled. And we could see that we had strong IBA1 staining from these hypertrophic and pro-inflammatory microglia in the tissues for those for which we'd seen these neuroinflammatory markers, suggesting that this really is a good, again, correlation between what's going on here in the RNA-seq data and what we can see going on at the protein level in these tissues as well. Okay, so this was published about 2 years ago by my lab. This is the summary of the subtypes. The ALS-TE group is the one we went in here looking for. And along the way, we found these 2 other subtypes of ALS, ALS-OX and ALS-glia, both of which are expressing markers of pathways known to be involved in disease, but which hadn't previously been appreciated would fall into separate groups. And I think that's one of the more interesting parts of this. So, okay, so what is my lab doing now? What are the big open questions that we're working on now? Well, you might have a few major questions from the stuff that I've been telling you about. One could be, well, how robust are these subtypes across clinical centers and in larger cohorts? If I take a random ALS patient, will they fall into one of these 3 groups, or are there more when you look in larger studies, right? Another question you could ask is, do these subtypes indicate cellular composition, maybe loss of neurons and gain of activated glia, for example? Or they could be representing disease stage. Maybe at the end point of the disease, you'll see different things from earlier on. or for example, maybe disease drivers. Maybe they really do have a different kind of ALS that's different from start to end, right? And then finally, are the transposable elements that I've been talking about just reporting on dysfunction? They're just reporting on TDP43 pathology, or could these actually be causal and driving toxicity in the disease? So that's what I'm going to tell you about in the last few minutes I have here. I'm going to start with the first one. How robust are the subtypes? So the first study was done with 178 samples. That was the 2019 release that was published a couple of years ago. We've been marching on with the sequencing study. We now have 500 samples from hundreds more patients. And what we can tell you is that there aren't any new groups that are arising when you add more and more patients to the study. They really do fall into these three subtypes, the ALS-TE, ALS-GLIA, and ALS-OX subtypes, and the relative distribution into these three subtypes is also fairly stable and similar, suggesting we had enough patients in the first study to actually reasonably describe what's going on and how many patients would be in each group. So this is work done by my student, Catherine O'Neill. She has since built a deep layer neural net classifier to take in any new sample and calculate its subtype and assign it to one of these three subtypes. That's what we've had to do, because it turns out that the original methods we use, non-negative matrix factorization, they have to run de novo clustering each time. They can't be run as a classifier in predict mode. And so the deep layer classifier that she's built has done very well and achieves better than 90% accuracy, both on the original assignments and the new ones that are coming in. And again, I'd be happy to talk more about that one later. But instead, I'm going to move to some of the other questions because I think there's really more interesting, cool biology we could be talking about here. So when answering this question about what the subtypes represent, we thought back to our original idea that we got here by following TDP43 targets and TDP43 pathways. And we knew that the ALS-TE subtype looks a lot like loss of TDP43. And in addition, some patients with an inheritable form of ALS carry mutations in TDP43 protein, but they're in TDP43 gene, but there are many, many other genes that are ALS associated, including, for example, TREM2. It's another gene for which mutations have been associated with neurodegenerative disorders, and specifically ones with a neuroinflammatory component. And again, we knew that the ALS-GLIA subtype looks in part like hyperactive TREM2 signaling, right? Either through these microglial stains or through TREM2 targets, which we could use to try and see if that correlated with the ALS-GLIA subtype. Right? So, but then the next thing we asked is, what about all of the other ALS-associated genes? Through GWAS studies and large clinical cohorts, we've now associated about 40 different genes with the familial form of ALS. Would each of those genes fit into one of these subtypes I've been describing, or is the genetic form of ALS really separate from the sporadic form? And so that's what we're going to ask next. And the way we did it is this. We've used that same idea of looking for the targets of an ALS-associated gene and asking if it was correlated with one of the particular subtypes. So for here, I'm just going to shorten this and talk about the ALS-TE subtype alone. We knew that targets of TDP43 were misregulated in this. So then we looked at knockdown experiments from almost any other ALS-associated gene where they'd either knocked down, knocked out, or knocked in the mutation, and asked what are the targets of that gene and do they correlate with this particular subtype. And what we found is there's multiple ALS genes that correlate with the ALS-TE subtype, including SMN1, survival of motor neuron 1, matron3, and to a certain extent, C9orf72. So then we broadened that to look at all of the subtypes. And what we could tell is that that they all split, right? And that most of the genes were either associated with the ALS-TE subgroup or with the ALS-OX subgroup, with CHCHD10 being the sort of prime candidate for that one. And then separately on top of that, other mutations would drive targets associated with neuroinflammatory processes, and that would mimic the things that were going on in the ALS-GLIA subtype, such as TREM2, which drives this one alone, or C9orf72, for example, which showed both signs of neuroinflammatory pathways as well as signs of TDP43 dysfunction, looking a little bit like both of these two groups. And so I think that lets us connect ALS genetics and the targets of these genes that we've been pointing to by these GWAS studies and the pathways that are really vulnerable in motor neurons and vulnerable in ALS as a disease. And so we're following up on this. Now, this does not mean that the other two things, cellular composition and disease stage, are not also involved. They almost certainly are. But I think we can also say that it's probably in part disease drivers that are being indicated by each of these different three subtypes. Right. Okay, so then I'm going to go to my final question, and then I'll be stopping very soon. So that last question was, are TEs contributing to the stress that kills neurons instead of just reporting on dysfunction in this particular disease? And so if you were going to think about that, if you were going to think about how TEs might cause cellular stress or cell death, there's a few ways by which that could happen, right? They could either be misregulating adjacent genes, like I told you about before. They could be activating pro-inflammatory pathways, perhaps even activating neighboring glia. That would be one possibility, right? And then another possibility would be that these could be actually inducing somatic mutations by hopping around genomes, and possibly even inducing apoptosis that way, right? So I'm going to tell you today just about this one. What is the evidence we have that transposons are associated with activation of microglia? Because remember, that was a separate subtype. In other words, could TEs be producing toxic or pro-inflammatory byproducts as a result of their activity? And if this is the case, then we need to get specific about the locus, because some transposons can encode proteins and others can't. So we need to know specifically which locus we're looking at. And then we need to get specific about cell type, right? So in other words, single cell data. Okay, so I'm going to talk about the locus-specific analysis first. This is hard for transposable elements because they're very sequence similar along the whole length, and there's many, many copies of them. And so the best way to address questions like that is to use long-read sequencing platforms like IsoSeq from PacBio or Oxford Nanopore to try and see whether or not you can tell exactly which loci is actually being expressed in cDNA samples. And so my lab has been adapting our algorithms to both generate those PacBio IsoSeq libraries and then adapt the algorithms to use that data and try and pick out exactly which locus is being expressed. And I'll show you just a little tiny bit of data towards that. So this is cells in which we have induced transposon expression by knockdown of TDP43. This is the cells, the loci that show upregulation in the Illumina sequencing data, and these are the loci that show upregulation in the PacBio IsoSeq data. And what you can tell is that we get pretty good correlation between which locus we predict is being expressed from those short read samples and which ones we actually measure to be expressed from the long read samples. And in particular, some of these loci are young enough to actually encode fully intact envelope proteins, which are the ones that have been shown to be neurotoxic in other studies. Okay, and then finally, in the last few minutes, I'm going to talk to you a little bit about single cell data. So how we're using this to try and track transposon genomics in single cells. Because if we're going to ask about cell autonomy and cell type, we need that kind of data as well. Right, so I'm going to go from that sort of cartoon version of what's going on to a slightly more descriptive version of what we're talking about when we're thinking about whether or not transposons could act as a source or a trigger for neuroinflammation. So what we're really talking about here, in the case of ALS, is that these TDP43 aggregates lead to upregulation of transposons. So they're desilenced. Once they're desilenced, they can start producing byproducts like double-stranded RNAs or cDNAs. Those will go through the innate immune sensing pathways, things like RIG-I or cGAS sting. And the prediction is that they would produce interferon as a result of that, and that would be secreted and could potentially activate neighboring glial cell types like astrocytes or microglia. That's the idea anyway for how this would occur. So just from the bulk data, we tried to see whether or not these components were actually present, whether or not this had a chance of being true. So what we did is we divided all of the samples in the ALS and FTD cohorts into ones that had the highest levels of this very young endogenous retrovirus called HERV-K. That's one of these very, very young transposons, right? And we also asked whether or not the innate immune sensors that we predict to be downstream of activation of this particular element were also highly expressed. And again, that was true. So MAVS is one of these RNA sensors, and IRF7, this interferon response factor, would be one of the things that we would be predicted to be upregulated if these pathways were active. And again, those were also the same samples that had markers of activated microglia, such as CD9, CSF1, and TREM2, as well as pro-inflammatory astrocytes, markers like C3, S100B, and GFAP, right? So, okay, that's bulk, but what about single cell now? Can we now tell which cells are actually expressing these things and what's active? And so for that, we need single cell data. So I'm going to pull a tiny bit of a bait and switch on you. I don't have the single cell data yet for ALS. We're getting it. So I'm going to show you a related disease where neuroinflammation is also a part of that neurodegenerative disease, and where transposons are proposed to be playing a role there, and that's Parkinson's disease. Right. So this is a single cell data set that we generated from substantia nigra, which is the affected tissue in transposon in Parkinson's disease. And we could build these lovely gene expression maps from standard single cell analysis software. But all of the software that's out there will quietly discard transposable element reads because they're a pain to map with for multiple processes during single cell analysis pipelines. Okay, and one of the reasons is that in nuke-seq data, which is what you need to do if you're looking at postmortem tissue, because you can't get the whole cell intact out, you'll have both expression from gene exons, that's what you're hoping is going to be one of the reads in your data. You'll have pre-processed pre-mRNA transcripts that are not quite fully processed yet and contain lots and lots of introns, and packed into those introns are transposable elements. And it can be very hard to tell the difference between a retained intron that encodes a transposon and a transposon just driving its own expression. But sometimes it really is the TE driving reads from the locus. And so we need to be able to do better with this software to figure out when it is reads coming from a transposon. And so again, we're going to use that same EM strategy that we used before in RNA-seq data, except now the components that we're going over are not just copies of TEs, but we're also going to add in host gene exons and host gene introns, which could be harboring transposable element sequences in them to this EM algorithm to try and figure out which one is actually generating reads from the locus, right? And then there's also, because some people are a little bit nervous about including transposon reads when there's many retained introns out there, there's a conservative version of the algorithm where we just look at intergenic loci and where we can't be fooling ourselves from reads that come from introns. And so I'm going to show you in the last minute, I have a little bit of data from that conservative method where we're looking at intergenic ones, right? So again, this is Parkinson's disease. Again, inflammation is a component, and we think transposons might be involved. This is work with Johan Roger Barker from Cambridge and Agneta Kirkeby, who's here at Lund as well as Copenhagen. And so we got single cell samples from the substantia nigra. And one of the things that we can tell, this is L1HS, one of those young transposons. There's plenty of transposon expression in these samples that can be gotten from single cell reads. And if we look and zoom in right in the astrocytes, one of the cell types we're most interested in, so GFAP marks all of them, we can see that transposons are most highly expressed right along this edge here, which is also where we see markers of activation, such as C3, which is one of those pro-inflammatory astrocyte markers, right? And that this edge is concentrated in Parkinson's patients and really not in controls, suggesting this is part of the disease process. And this pattern of significant upregulation and of transposable elements, as well as correlation with pro-inflammatory markers, is seen for multiple transposable elements, consistent with this idea that they tend to be all co-regulated together. I'm going to skip the microglia stuff and just say one last thing, which is we still need to, of course, validate this. I think we've now got tools to look at this and to predict where transposons might be active, and the rest of my collaborators are going to be doing some really heroic work to try and upregulate transposons in each of these cell types and ask whether or not they are sufficient on their own to induce neuroinflammatory pathways. And I'm really excited, hopefully, to tell you more about those results later. Okay, and then I want to thank my lab who contributed to all this work, all of my collaborators, and you for inviting me to give a talk here today. Thanks so much. Yes, I can. Yeah. Yes, that's an excellent question. So one of the things I skipped over a little bit is that each one of us probably has at least 100 to 1,000 different loci in the genome where my transposons differ from yours. And those differences are generally presence absence. I might have a transposon at that particular location and you don't, for example, right? And we've just really been getting better at building larger, long-read, well-assembled genomes from those like UK10K and 1,000 genome projects to try and understand better how much polymorphism there is both in these sort of presence absence as well as SNPs along the lengths of the transposons. That data really isn't being incorporated yet, but I think it should be, especially in these cases where we're interested in whether or not patient populations might be more likely to have a copy that's a little more dangerous than the rest of the population, for example. And so I think that's a really interesting question and possibility. And I hope that at some point we're all going to move towards graph genome representations that better take into account those sorts of polymorphisms when we're doing mapping to studies like this. Mm-hmm. Yeah. But that's an excellent question. We are doing that EQTL analysis. Oh, that's my timer telling me I need to stop. We are doing that EQTL analysis to try and figure out whether or not any of the genetic loci are actually associated with either, polymorphic transposons themselves or regulators of transposon expression. I don't have any results from that yet, but I think that's a really interesting idea and something we're certainly very interested in doing. Again.